So far, we've examined gene trees and how gene trees might not match up to species trees, and that's due to incomplete sorting of alleles. And how we then looked at how molecular phylogenies can be used to answer various questions in terms of the evolution of certain traits or lineages. Here, we are coming back to the topic of natural selection and neutral evolution and seeing how we can use neutral evolution to estimate how long ago certain events happened or how long ago lineages split up from a common ancestor. So before we get to that concept, let's first reiterate that a lot of our non-coding DNA, such as pseudogenes, don't have any known function. So pseudogenes don't do anything. They don't code for anything. So hence they're not likely to affect phenotypes at all. They're not likely to be exposed to natural selection at all. If nothing is being expressed, then nothing can be beneficial or selected against. Also, in the same realm of neutral sorts of evolution, synonymous or silent mutations in protein coding genes do not have any effect on a protein by definition. They might be changing, or they are changing, a nitrogenous base, but the same amino acid is used in the protein, so those are going to be neutral. And then finally, some amino acid substitutions are called nonsynonymous, or you might know them as missense mutations. And in those, a different amino acid is used in a protein. However, if the amino acid has a similar chemical characteristic to it, like in its charge, then it may also not alter the function of a protein and may be neutral. So the overall point in this first section is to lay out the groundwork and understanding that there are numerous ways in which the genetic makeup of an organism can be neutral. So way back in 1986, Kimura argued that much of the genetic variation we find in the genomes of organisms is the result of genetic drift. And so he was basing that on his argument that much of our variation in various genomes is neutral. And if it's neutral, therefore selection can't touch it. Only genetic drift can randomly cause it to change frequency. A prediction of this neutral model is that neutral mutations should become fixed in a population at a regular rate, a clock-like rate, if things are happening neutrally and nothing is being positively or negatively selected for or against, then the clock-like appearance of mutations coupled with the random wandering from genetic drift should result in mutations becoming fixed at 100% at a regular clock-like rate. To test this prediction, Fitch and Langley, several decades ago, obtained cytochrome C sequences from a variety of different mammals and plotted them, the differences in the sequences, the number of substitutions in cytochrome C, plotted that against the amount of time since these various types of mammals last shared a common ancestor. And we know that from the fossil record. So we get the genetic data from our friendly geneticists. We get the fossil data from our friendly paleontologists, and we do a collaborative project. And what you can see in the graph on this slide is it's pretty remarkable how linear it is. The number of substitutions in the cytochrome C gene has been relatively clock-like and constant over time when plotted against how long ago common ancestors were shared according to the fossil record. And this relatively constant rate of genetic change that's presumably due to neutral variation is referred to as a molecular clock, and it's used to date how long ago lineages split from common ancestors. So let's take a look on the next slide, besides this application right here with mammals, of how this molecular clock approach was used to estimate when HIV-1 jumped into humans. So before we get to the HIV-1 example of using a molecular clock, let's first take a pause and get a sense, get an understanding of how different types of genetic changes occur at different rates, slow and fast rates. The rate of nucleotide substitution per site per billion years. And what you can see from this graph is that the rate of nucleotide substitution is relatively low for non-synonymous sites, that is, sites that are giving rise to different amino acids and proteins that can result in a different function. Synonymous sites are farther over on the right in this scale, and they have a much faster rate of substitution. Introns, which are non-coding, have an even faster rate of substitution, and then pseudogenes you can see is the fastest. So basically what we're seeing is those parts of the genome that are increasingly neutral in their effect evolve at a faster rate because genetic drift is able to allow them to change, where a selection would perhaps, like with non-synonymous sites, keep them relatively conserved. So pseudogenes evolve at a far faster rate than non-synonymous sites, and neutral theory is supported by several lines of evidence like these. So here's an application of the molecular clock used to estimate the origin of HIV-1. So, of course, HIV was first being detected in the 1980s, and from then on it was, known to science and the public. So to get an estimate of when HIV-1 may have originated, biologists compared RNA sequences from 159 HIV-1 viruses collected from different patients over the period of 20 years. And based on the rate of mutation, the mutation rate over this 20-year period, they're able to sort of back calculate using the molecular clock approach. And they estimated that HIV-1 must have jumped over to humans from our primate relatives between 1915 and 1941. So that's quite a time range, and it would be great if we could narrow that down. That range existed because of the limited amount of data we have over a 20-year period for HIV. Well, biologists were able to get some older blood samples that existed in, for example, the Democratic Republic of the Congo, and that those older blood samples that also contained HIV. And so they're able to get a more precise estimate by looking at the mutations in those older blood samples, and including those, we get a more precise estimate of the early 1900s using that molecular clock. So that's what the graph here is showing you. Now, interestingly, this estimate for when HIV-1 jumped to humans, early 1900s, coincides with times when there were new human settlements taking place. And these new human settlements meant bigger population sizes in areas that are intermixing with wildlife and collecting, say, primates for food, and having the chance of mixing blood between species of primate and human. And so these settlements probably increase the chances of infection crossover from some other primate to human. So, okay, molecular clock, it works. Now the next section is titled Footprints of Selection. And the idea here is that there are several methods that biologists have developed to be able to determine periods of natural selection that occurred in the distant past. Selection that we're not measuring today in current populations, but selection that we logically and reasonably conclude must have occurred in species in the distant past. So how in the world could we do this? How could we know that selection operated on certain genes in the past? Well, as I said, there's several methods that biologists have developed, and I'm not going to cover all of those in this section, all the ones in your book, time reasons, but let's look at two of them. The one here is using linkage disequilibrium. So neutral mutations can take a very long time to reach a high frequency by genetic drift. So variations can bop around and jump around randomly, and especially in big populations where drift isn't very powerful, it could take a very long time for a particular allele to randomly get to fixation by drift. Now, over long periods of time, if a mutation pops up, genetic recombination through crossing over, etc., can mix and match surrounding stretches of chromosomes generation after generation. And if a mutation pops up, that erosion of linkages is going to happen, and we won't over time have certain alleles that are tightly genetically connected to that new mutation because they've been broken up by genetic recombination. What happens though if a mutation is favored by selection and there is what's called a selective sweep that occurs? Then we can detect that. We can detect that because those alleles that are nearby the mutant allele, if selection was very strong and pushed a favorable allele to fixation pretty quickly, then there would not be enough time for genetic recombination to break up the surrounding alleles from that new mutant. And we can expect to see relatively little genetic variation around those selected mutant alleles. This is what's being shown in the figure that's on your slide. So in figure A, it's showing you the horizontal lines are individual organisms, and the little colored dots are showing alleles that are found in these organisms. So the star shows a mutation that occurs at that site in an organism, and that mutation represents an adaptive allele that came into existence. Now, if a selective sweep occurs and that allele is pushed to fixation relatively quickly, then what we'd expect to see is any nearby allele, like the light blue one that's there near the star, is not going to be broken up from genetic recombination before that allele is pushed to fixation. And so we'll see in many individuals that have that mutant that has been selected, we'll see a similar nearby allele close by, in this case the light blue one, and that shows positive selection. The figure C that's on the bottom there is showing you that when we scan across a genome, we can detect a selective sweep as a valley in the amount of genetic variation. And so basically this is along the same lines of what I've just explained. If you're looking through a genome and you see all of a sudden in a particular region, oh my gosh, there's hardly any genetic variation. That could be a signal that there was a selective sweep and there is an allele there that was under positive selection and went to fixation, and hence genetic recombination didn't break up the surrounding alleles that are associated with that. Every individual organism has inherited that beneficial mutant along with the surrounding alleles that haven't been broken up. And there we go. There's a signal. There's a signal for a selective sweep. Low amounts of variation in certain regions of the genome. So this is one way. Linkage disequilibrium can decrease over time, but if there's a selective sweep, then that linkage disequilibrium will still exist, and we can detect that by finding regions in the genome that have relatively little variation. So one neat example to share with you here about how we can use linkage disequilibrium to detect a selective sweep for a particular mutant is with lactose tolerance in humans. So as you probably know, most mammals, when they are weaned as a baby from their mother and drinking milk, they start feeding on the normal adult diet, whether it be plants or meat or whatever. For mammals, they switch off the production of lactase, so those adult mammals are no longer able to hydrolyze or digest lactose as a sugar source. There's no need to. But in humans, about 30% of adult humans are actually lactose tolerant, which is very unusual for mammals. So first of all, what's being shown here on the bottom of the slide is that areas where humans have the greatest percentage of lactose tolerance, those areas are tightly associated with regions where cattle were domesticated several thousand years ago and used as a dairy source for food. So that's indirect evidence to help pinpoint regions where these lactase mutants came about. The highest frequency of that lactase, those lactase mutations, are in areas where cattle were domesticated and used as a food source. Again, that's the map on the bottom. But we also have much more persuasive and stronger evidence that a selective sweep happened several thousand years ago, favoring those lactase mutants and pushing them to fixation relatively quickly. And when that fixation was pushed pretty quickly, again, recombination was not able to break up those alleles nearby the mutants. And so there's relatively little genetic variation when we compare different individuals around those mutations. If you look at the figure on the right-hand side of the slide, the top figure is showing you results from Africa, and the bottom figure is showing you results from Eurasia. So what you see on the top figure is those individuals that have the lactase mutation that allows them to be lactose tolerant into adulthood. They show very little variation in their alleles surrounding that mutation. And those are the long blue lines that you see on the top of the figure. Whereas individuals who are lactose intolerant and do not have that mutation, those are the purple lines down below in figure A, they have sexual recombination has broken up those linkages, and those individuals show very high amounts of variation around their lactase alleles. So there's one piece of evidence right there from African populations, but we see the same pattern, a different study, showed the same pattern among Eurasian people. And so once again, the orange lines show individuals who are lactose tolerant and carry that mutant lactase allele, and they have relatively little allelic variation in genes surrounding that mutation. Those are the orange lines. Whereas individuals in Eurasia that are lactose intolerant, they have high amounts of allelic variation around their lactase alleles, and those are the purplish lines down on the bottom. So we can look for signals of selective sweeps by looking at the degree of linkage disequilibrium and using that as evidence for alleles that were pushed to fixation relatively quickly by natural selection. So here's the other example I wanted to share with you for how scientists, biologists can detect potential selective sweeps and demonstrate whether mutations were subjected to selection or not in the past. So here, this is titled Footprints of Selection, Changes in non-synonymous mutations compared to changes in synonymous mutations. Okay, so that's the DN over DS. Now, neutral theory is used as a null hypothesis for evolution. We're going to assume that evolution has occurred by random genetic drift neutrally unless there is evidence to the contrary that suggests natural selection. This is very similar to the approach used with the Hardy-Weinberg equilibrium. We assume that there's been no evolution in a population whatsoever unless we can detect that the population is out of genetic equilibrium, in which case we know that one of those evolutionary mechanisms must be responsible, like selection or drift or gene flow. So it's a very similar kind of model that we're using. Genetic drift and neutral theory is the null hypothesis, and accepting an alternative hypothesis of natural selection is only going to happen if we have evidence to reject that neutral theory. Okay, so what we can do is we can compare synonymous and non-synonymous substitutions and look at the ratio of them in a particular gene. And based on the ratio of them, this gives us a signal as to whether there may have been positive neutral drift, positive selection that is favoring a particular mutant, or purifying selection, which is getting rid of new mutants. Okay, so in figure A, what we're seeing is, okay, we've got a segment of DNA, and it's got the nitrogenous bases adenine, cytosine, thymine. And that sequence, that codon, codes for threonine. Okay, fine. So if you have a synonymous mutation and that third position, the thymine, is changed and substituted with a cytosine, that's on the left-hand side of the figure, you're still coding for threonine. So that is a synonymous mutation, substitution, still have threonine. On the other side of the flow chart there, a non-synonymous substitution, changing that first base adenine to a guanine. Okay, now that doesn't code for threonine anymore, that now codes for alanine. That's a non-synonymous mutation. So that part of the figure is just clarifying and showing you what would be a synonymous versus a non-synonymous mutation. Now, if we compare then the number of synonymous and non-synonymous substitutions, the latter of which is subjected to natural selection, non-synonymous ones, because new amino acids are put in and that could alter the shape and function of a protein. So if there is neutral drift and selection is not involved at all, then we would expect that changes in non-synonymous substitutions is going to be equal to synonymous ones. And so that's the top figure up on the upper right. Is if we look at the pattern of mutations in a lineage and we find that the pattern is basically equal, there's an equal number of synonymous and non-synonymous ones, neutral drift is involved. If we look at the pattern of evolution of mutations and we find that non-synonymous ones are much more frequent than synonymous ones, that would be the middle figure, then that would be an argument for positive selection, that those mutations were favored by natural selection and non-synonymous ones were pushed to a higher frequency. And then the final scenario, which is on the bottom of that figure, purifying selection, if we find that changes in synonymous substitutions outnumber non-synonymous ones, then that is evidence for purifying selection. That is, those mutations are being weeded out because they have a detrimental effect on the organism, and the status quo sequence is maintained. So that's purifying selection, and that's when synonymous mutations we find to be more common than non-synonymous ones. So very interesting, useful tool, comparing non-synonymous and synonymous mutations to be able to detect whether there's been neutral drift, whether there's been positive selection, or whether there's been purifying selection. There are other methods that your textbook talks about for detecting selection in genes, but I'm just not reviewing them because of our short amount of time and an enormous amount of information to cover from this book. All right, so we're nearing the end of the chapter here and what we're covering, and remember that the chapter is called The History in Our Genes. Our genes are telling us something about past history. So this last section is titled Genome Evolution, the evolution of whole entire genomes. And what's being pointed out here is that in bacteria, the size of the genome is directly proportional to the number of genes in each species. There's very little, non-coding DNA in bacteria. So gene duplications and horizontal gene transfer will increase the size of a genome, whereas deletions are going to decrease the size of a genome as long as fitness isn't reduced when a deletion happens. And as we'll see on the final slide, symbionts, those bacteria that live with other organisms that are their hosts, they tend to have smaller genomes, and we have a good idea of why. So that upper graph is showing you the very clear message that in bacteria, genome size, genome length, is tightly associated with the number of protein coding genes. So there's a clear relationship. But when it comes to eukaryotes, look in the bottom figure, there is not a clear correlation between genome size and the number of genes. And quite frankly, biologists do not know exactly why. There are some hypotheses out there, but we have yet to really nail down why. So you can see genome length in megabytes and megabases, and ranging from 0 up to 3,000 on the x-axis for eukaryotes, and the number of protein coding genes on the y-axis. And you can see that it's more or less a scatter plot. It's all over the place. So there is no clear association in eukaryotes between genome size and the number of protein coding genes. So that remains to be figured out. But the last slide coming up, let's take a look at some of the reasons why in bacteria genome size varies according to their niche, how they live. Let's check it out. So here we go, reduction in bacterial genome size. We see this pattern over and over again, so it's pretty clear that this relationship between genome size, and where and how the bacteria live is a pretty strong relationship. So it's related to niche. And this figure is really speaking loudly to what's going on. So take a close look at the figure. But basically on the left-hand side of the figure, you're seeing, okay, for those bacteria that are free-living, they don't live with a host, they don't live closely with some other organism like a plant or a fungus or an animal or whatever, then those bacteria tend to have relatively large genomes. And that suits them because free-living bacteria live in environments that are environmentally much more variable. And so it's going to suit such bacteria to have larger genomes to cope with the range of environmental variables they might be exposed to. So they tend to have relatively few pseudogenes. They don't have very many mobile elements jumping around. And they have ongoing gene acquisition and loss. So they acquire new genes by mutation or horizontal transfer, loss through deletions, etc. But working towards the right in our spectrum of possibilities, if there's a bacterium that's a relatively recent show up to being a symbiont with some host, so a relationship with some kind of symbiont hasn't existed for very long, then we see a slightly smaller genome. And we see quite a few mobile elements, quite a few pseudogenes. We see both large and small deletions. So we see smaller genomes, but there's still quite a bit of activity going on with those genomes as those recently evolved symbionts are going through transitions. Then as you continue to go right on our graph, we have relatively long-term obligate symbionts or pathogens. So they have a long-term relationship with the same kinds of organisms. Their environment is relatively stable, and you can see their genomes are even smaller. And that pattern continues all the way to the right side of the figure with the tiniest genomes of bacteria that have the tightest, most long-term symbiotic relationships with hosts. So there's a clear relationship between genome size and how bacteria are living, whether in free-living environments or with different long-term relationships with symbionts.